So I mentioned that the chromosomes are packaged up in the preparation for cell division. And cell division would be a smaller part of the overall, what we call the cell cycle. So this is an illustration of the cell cycle. This is showing yeast, the budding yeast, which is a kind of a model organism for studying cell division. So we start with a cell, small cell gets larger. One of the phases here, this is G1, which is the first growth or gap phase. I've heard the G standing for either of those. And nothing really happens with the DNA at that stage, but the cell gets bigger. And the S phase is an important phase. This is the process of copying the chromosomes. So I know I mentioned that the chromosomes are not always packaged up like this, but this is the easiest way to think about them. So they are showing here with our chromosome number, and that chromosome number is 1, 2, 3, 4, 5, 6, 7, 8. So this is a cell with 8 chromosomes, again, just kind of hypothetical. And the fact that the chromosomes exist as pairs, I've got them colored for, say, one from mom, one from dad. The fact that they're paired, that tells us that this is a diploid cell. So this is a chromosome number you would call a 2N for diploid. 2N is 8. See if I can keep doing this. So after this S phase here, then the chromosomes are replicated. So S is stands for DNA synthesis. And that's basically the same thing as DNA replication. So the DNA gets copied. So we talked about in dividing a cell, you've got to have twice as much DNA so that one set can be put into one cell and one set can be put into the other cell. So one question that always throws people is, what is the chromosome number after synthesis? So we started with 2N is 8, and now we've got 1, 2, 3, 4, 5, 8. So after DNA replication, the chromosome number is still 8. So we have 1 pair, 2 pairs, 3 pairs, 4 pairs, 1 pair, 2 pairs, 3 pairs, 4 pairs. It's the same number of chromosomes. And the difference is that each of these chromosomes has 2 halves. And I use a terminology that I like to use called, referring to these as replicated. So these are replicated chromosomes. They're chromosomes after the process of replication. These are chromosomes that would be referred to as unreplicated. So we have twice as much DNA as a result of DNA synthesis, but it's still the same number of chromosomes. So when I refer to a chromosome, I'm talking about something that is physically united. We often draw them in this X shape, but they're physically connected at the middle. And so this whole thing counts as one chromosome. So referring to this as a replicated chromosome and this as an unreplicated chromosome allows us to distinguish them. All right, so moving through the old cell cycle, after these chromosomes have been replicated, then they go through this process of, so there's another growth or gap phase, G2, and then there's this process of mitosis. So M is mitosis. And that's the division of the nucleus. Nuclear DNA divided into 2 nuclei. So I'll mention a few times during this class, I just smush it all together. A lot of people like to remind you that mitosis and cell division are different things. I'm going to think about them in this class as basically being the same thing. Or another way to look at that is that mitosis leads to cell division. So copying the DNA and then dividing it into cells. Dividing is dividing into nuclei and then dividing into separate cells. So at the end of that, we've got the arrow split here. This newish cell that was made has 1, 2, 3, 4. 2n equals 8 chromosomes. As a result of splitting, these are now unreplicated chromosomes. And this cell then has DNA that looks just like the DNA of the starting cell. And then we can leave that on there, and that would be the DNA of this other cell that results from the cell division. And that can go on to continue the cycle. So every time around the cycle, we get another cell, and each cell that leaves this cycle has a full set of DNA. So this is important to be able to maintain a cell, right, going forward. Double the DNA, split the cell, double the DNA, split the cell, and that's the process. And so every time a new cell is made, it has the same essential components of being a cell as the cell that started it. So in this diagram, all the cells are diploid. Nothing is ever haploid. And that chromosome number, as we saw, is 8. 2n equals 8 all throughout. Okay, so I mentioned replicated chromosomes, unreplicated chromosomes, a few more chromosome terms on here. The centromere. I don't enforce these terms too much in this class. Again, you've probably had them before. Certainly important to processes that involve them, but not super important to general genetics in my mind. So centromere is this kind of pinched together part on an unreplicated chromosome. I usually draw it just sort of pinched in. On a replicated chromosome, the centromere is what holds these two halves together. The technical term for those two halves are sister chromatids. Chromatids, sometimes people like this term a little bit better. So chromatid, I find it sometimes a little confusing, but whatever a student's comfortable with, really. So chromatid is basically like one of these things start to finish. So this is one set of one copy of chromosome. And so an unreplicated chromosome consists of one chromatid. A replicated chromosome consists of 2 chromatids, and we call them sister chromatids. And you could even extend that a little bit further to say that they're identical sister chromatids. So these are the result of copying. And so the left-hand side and the right-hand side should be exactly the same, barring any mistakes that were made during the copying. Another term on here, the telomere. These are important end parts of these linear chromosomes, and they help to keep them intact over time. Oops. So I mentioned the centromere. The centromere has an important job. It's a little bit outside the scope of this course, in my mind, but it's kind of a cool thing to think about. The centromeres are distinctive pieces of DNA that interact with proteins that are involved in the two parts of basically keeping them together when they need to be kept together and pulling them apart when they need to be pulled apart. So the left-hand image, I like this quite a bit. While they're together, you can see there's this kind of Velcro detachment that's being depicted there. They're held together on a molecular level. So the proteins of one chromatid are keeping that anchored to the other chromatid. But also during the process of mitosis, there are these microtubules heading off to the sides, and those are pulling the chromosomes apart. So for a while there, the chromosomes or the chromatids are stuck together, and they're being pulled by the tension of the microtubules. And what's being shown in this video is the break point when the chromosomes split, where the two chromatids that are held together are no longer held together, and they're allowed to move to the opposite sides of the cell. So a really nicely orchestrated, elegant process in a cell to be able to pull those apart. On the right-hand side here, you can kind of see the maybe the organization of those. It's showing a lot of the process that's after the division. So I don't know if that really quite shows it. But it does show them being pulled. You can kind of see these floppy chromosomes being pulled by the circular, the dots that are put on there indicate the positions of the centromeres. And so those are where these fat noodles are being pulled apart. All right. So more detail than you need to know, but maybe you're curious about these. There's, again, distinctive sequences of DNA that are recognized by specific proteins. And those proteins have to hold on for a certain period of time, and they have to let go also at the right time. So they're attached to the DNA of the chromosome, but also attached through a variety of these proteins with the microtubule that's going to do the pulling. Cool stuff, more than we, I think, can handle in this class. All right, so diploid cell, just like we saw in the cell cycle illustration, a diploid would have two chromosomes that are homologous. Sometimes you see them colored like this to tell you where dad's contribution and mom's contribution are. As a pair, they're homologous, but homologous chromosomes, for the most part, don't interact with each other. So we can line them up artificially, but they're not necessarily going to be friendly with each other just on the average day in the cell. So going from unreplicated to replicated, we still have two chromosomes. They're still homologous, just twice as much DNA. So you can kind of go between these two stations of replicated and unreplicated. The process of going from unreplicated to replicated is the process of DNA replication, go figure. And the process that gets you back to unreplicated, that's the process of mitosis, where these replicated chromosomes are broken in half, and each half goes into a new nucleus, a new cell, as we've seen. All right, so let's try to get it back to genes and alleles for a while here. So if you have a gene in a cell, the gene is located at a specific place on a specific chromosome. So one of the terms that's sometimes used for a gene is we refer to it as a locus, kind of a fancier way of talking about a gene. And locus is literally the Latin word for place, or you could say position. So if you have a gene, let's say you have the A gene, then wherever it's located on this chromosome should be the same place that it's located on this chromosome. So same gene, could have different alleles for that gene, but should be found at the same place in almost all members of the species. All right, so here's an example. Let's say we have a gene that does have differences that are alleles. During the process of replication, where does big B and little b, where do those end up? So I think the color coding helps here. So if we start with little b on the bluish chromosome, big B on the orangish chromosome, then by copying blue, we get B on the same chromosome, sorry, little b on the same chromosome as the little b, and a big B on the same chromosome as the big B. All right, these two halves are identical. All right, so we kind of did that one, but if you want to quiz yourself on how these would look, you can come back and try this one again. So I don't cover too much detail when it comes to mitosis, but this is an illustration of the process. Again, I think you've had it before in other classes. I'm interested in the genetic result of mitosis. So sometimes it's important to follow chromosomes. Let's say you've got genes on the same or different chromosomes, that would be important. But really it's a question of, okay, what's going to end up in the resulting cell genetically? And we'll do an example of that in a little bit here. Just a couple notes about this process. So mitosis, technically, as I mentioned, is the process of dividing the nucleus. The fancy term for cell division is cytokinesis. And for my money, for this class, we just kind of squish it all together. So mitosis plus cytokinesis is part of the cell cycle. Before that, as we saw before, Very important, this S phase. When I was first learning mitosis and meiosis, I think I kind of overlooked the importance of the S phase. So if you take your ordinary cell with 4 unreplicated chromosomes, you can't go with this cell and start mitosis or meiosis. You've got to first copy the chromosomes. So keep that in mind every time. You got to start mitosis and meiosis with replicated chromosomes. And as we'll see here, the process of mitosis involves taking the chromosomes that exist in a cell and dividing them equally so the resulting cells look like the starting cells. So here we've got short blue, short red, long red in the starting cell. The resulting cells get short blue, short red, long red, short blue, short red, long red. So they each get the same kind of DNA. This last question here, how do cells ensure that DNA is distributed equally? That gets me to basically my favorite part of mitosis is this phase called metaphase. So we'll do some diagram of metaphase. During that phase, you can really see how this lineup of chromosomes is going to cause half of the information to go to the left and an exact copy of that information to go to the right. So I don't need you to know prophase, metaphase, anaphase, all of those, but I would probably ask you about metaphase. You don't have to know all the phases, but keep this one in mind. So back to this question, what situations require exact copies? So a cell making an exact copy of itself. If you're a single-celled yeast cell, this is how you divide and take over your environment. So it's a, sort of a form of replication by making exact copies, asexual replication. And in a multicellular organism, this is how individuals grow. And this is also involved in wound repair or just sort of cell replacement over time. So it's a process where the genetics or the integrity of the organism stays the same, but it might have more cells at the end of this process. All right, so let's try and diagram one of these. Let's say we have these chromosomes with these genes on them, with these alleles. What happens to them in a cell? What would happen during metaphase? So let me see if I can copy them over. No. Here we go. All right, so we had a couple of big chromosomes and a couple of smaller chromosomes. So this is a 2n equals 4 situation. All right, so little q, little t. And that must be big Q and big T. These are all supposed to be the same size. Sometimes I get my drawings a little bit off. Big R, little n. And little r, big N. Try to keep my handwriting clean on this one. And we'll see what happens. Okay, so what's been shown is the unreplicated version of these chromosomes, right? So we've got 2 unreplicated large chromosomes, 2 unreplicated small chromosomes. So important first step is to do DNA synthesis or replication or S phase. Replication. So just as we've seen before, by copying a chromosome, we get the same genes with the same alleles in every resulting chromosome. So if we had little q and little t chromosome, then on this same chromosome after replication, it would copy and produce little q and little t. Big Q, big Q, big T, big T, big R, little n, big R, little n, little r, big N, big N. So we've just made exact copies of these chromosomes. There's still 2n equals 4. And if you want to set up now for metaphase, some more space here. There's a lot more space here. To scale this down a little bit. So I'll draw them nice and spaced out. In reality, they might be a lot closer together. I can put them in the same order. I can put them in a different order. It doesn't matter what order the chromosomes are in here, because the left half of what I've drawn should be the same as the right half from a allele perspective. But I think I'll just keep things as familiar as I can for now and keep all the same genetics in the same order as we drew. So physically, these chromosomes are going to get pulled apart. So the left hand is going to go to the left side, the right hand is going to go to the right side. Maybe if we go up here to the starting point. So we have 4 genes and they're all heterozygous. So whatever order you want to put these in, just put a simpler genotype on there. So big Q, big T, little t, big R, big N, little n. This would be the genotype of the starting cell. And so QTRN, the resulting cells get 1 big Q, 1 little q, 1 big T, 1 little t, 1 big R, 1 little r, 1 big N, 1 little n, right? So 1 Q, little q, big T, little t, big R, big N, little n. So that would give you a full genetic complement for the right-hand cell that results from this. Left-hand cell, big Q, big T, little t, big R, big N, little n. So they'd be the same, right? You appreciate that? Cool. So you should be able to do this with chromosomes that I give you or describe to you, alleles, really any number of alleles. You should be able to do this. And a nice illustration that mitosis results in genetically the same thing as you started, just twice as many cells.